who is maize pathology leader in Sub-Saharan Africa, Summit Center. I welcome him and grateful to him for accepting our invitation to deliver the lecture. Thank you, sir. Now I request Dr. K.S. Huda, sir, Principal Scientist, ICR, NBPGR, New Delhi, for his welcome address. Huda, sir.
Yeah. Good morning, ma'am. Dr. Suresh, my team. I welcome Dr. Suresh, a renowned scientist working on maize. Today, he will speak on Mesolithic Necrosis, MLN, efforts towards containing the spread and impact of a devastating transboundary disease in Sub-Saharan Africa. Very important lectures to protect the countries and the world maize from this devastating disease. He is doing a wonderful job, and all will be benefited with these lectures. And I request Dr. Prashant Jambulkar to introduce the speaker to the audience.
Thank you, sir. Thank you, sir. Yeah. Maize pathologists around the globe, including me, know Dr. Alan Suresh, but still, I feel for the audience, we will customarily we will introduce Dr. Alan Suresh. Dr. Suresh is presently working as a maize pathology leader in Sub-Saharan Africa Summit Center, ICRF, Nairobi, Kenya. He has also worked as a plant health application leader, India, 2011 to 14 in Monsanto. He was an Asia lead in plant pathology applications. He worked in West Asia, Seminis, vegetable seed. He worked as a plant pathologist in Nunhems. He worked in Indo-American hybrid seeds. He has a vast experience with the private companies, multinational private companies too. His main research area are disease diagnosis, host plant resistance, host plant pathogen interaction, then epidemiology, disease surveillance, seed health, molecular diagnostics, exchange of germplasm, obviously disease-free. He has, for his research achievement, he has been honored by many honors and awards. And very recently, he has been awarded with the Hind Ratan Award by NRI Welfare Society India in 2021. He has also been awarded as a Scientist of the Year by National Environmental Science Academy India. He was also a recipient of M.J. Narasimhan Award, which is the highest academic award from the in the age of when he must be a student. He has also been appreciated by CIMMYC, Monsanto, and many a times. Means we are very much happy to have this acclaimed speaker with us. And now I request Dr. Alan Suresh sir to begin his talk. Sir, please. Please share your slides, sir.
Yeah, so I'll share my screen. Can you see my screen?
Yes, I think it should be in slide mode.
Yeah, one second.
Yes, not
okay.
It's fine.
Okay, thank you, Dr. Prashant, Dr. Huda, and for giving me a nice introduction. And I sincerely thank Indian Phytopathology Society for giving me an opportunity to present my efforts on maize lethal necrosis the effort done by CIMMYT and the many partners. And it is a great opportunity for us to give this entire updates to the many pathology colleagues and also students in the Indian community, as well as the many other colleagues in the world. So just a second. So now are you able to see my screen?
Yes, sir.
Okay, so you can see the main screen in the full screen mode?
Yes.
Yes.
So, and then also I thank Dr. Gugay and Dr. Pratibha Madam and for considering this lecture is one of the key topic for the entire pathology colleagues in India. And this is one of the most important transboundary disease and also invasive pathogen in sub-Saharan Africa. As you all know that maize is the most important cereal crop in sub-Saharan covering around 35 million hectares, largely in small holder farming systems that produces over 70 million metric tons of grains. And the crop is critical for food security, income, and livelihood of the many million farmers. Especially in Eastern Africa, it is mostly used as a food. And in the entire region, the maize is grown to the tune of 1.7 tons per hectare. Mainly the poor yield due to several constraints, especially drought, intermittent drought, poor soil conditions, poor soil fertility, inadequate use of inputs, and also the major is the pest and disease occurrences. The pest is the fall armyworm, which you are aware about it, and diseases such as maize lethal necrosis is the recent history, and previously maize streak viruses, gray leaf spot, and other diseases which I will explain you in a minute. So maize lethal necrosis is one of the most complex viral disease emerged as a serious threat to the maize production. And this is primarily due to the introduction of maize chlorotic mottle virus in the regions during 2011. Although the partyviruses, they were present in the region for several decades, but alone partyvirus did not make such a major damage. When the maize chlorotic mottle virus was introduced during 2011, due to the combined infection of maize chlorotic mottle viruses and sugarcane mosaic virus, that is one of the partyvirus, it has really affected the crop and really brought the crop almost 100% loss. So today I'll be covering about the major outlines are included in this lecture is that maize lethal necrosis, about the disease, how the global occurrence and distribution of MLN or MCMV, the disease resistance program has been done from CIMMYT, that is one of the flagship from CIMMYT, and how we put our effort on safe seed movement and with effective quarantine measures. Normally CIMMYT is known to contribute to the germplasms across the globe. But in this case, CIMMYT has done extraordinary effort in collaboration with various partners in Africa and also collaboration with other international research institutions in USA, Europe, and some of the leading institutions in Africa. So together with their support, we have done extraordinary efforts to contain and prevent the spread of this disease through safe seed movement and with effective quarantine measures. And also I will touch upon how we did our efforts on MLN diagnosis and surveillance, and how we did our efforts to support smallholder or small and medium enterprise seed companies to produce disease-free commercial seeds. And also we strengthen the capacity of partners in terms of disease surveillance and diagnosis. And we also developed a single information portal where all the information what I'm talking and which has been available in the globe is available in this web portal. And we also supported the community of practices. As you all know that in Africa, maize just is not a food, it is a life. It's everybody depends on the maize and almost 85%. So as with this, I was mentioning earlier that the maize is being affected by various diseases, especially fungal diseases. The main fungal diseases are Tuscicum leaf blight, grave leaf spot, Fusarium ear rot, but there are many pathogens of ear rot is associated, Diplodia, and several other fungal pathogens. But in this case, Fusarium ear rot is one of the key pathogen affecting the ear. And common rust, the main viral diseases is Mesolithic necrosis and maize streak virus. So majority of these viruses can be tackled only through having resistant hybrids or resistance breeding program in the companies or in the institutions. So as I was mentioning to you that this particular Mesolithic necrosis is a multi-pronged approach where we engaged multidisciplines in terms of disease diagnosis, in terms of breeding, in terms of molecular breeding, in terms of various seed production agencies, and several such organizations, and multi-institutions. It is not one institution, several institutions such as National Plant Protection Organizations, National Agriculture Research Organizations, small and medium enterprise seed companies, international research organizations such as CIMMYT, CABI, as such organizations, ICRA, AGRA, IATF, several such institutions together put efforts to develop a strategy to curb and prevent the spread of Mesolithic necrosis in sub-Saharan Africa. So this effort has been done through intensive germplasm screening, a fast-track development of deployment of MLN tolerant or resistant hybrids, especially using African adapted genetic backgrounds. So we also focused on optimizing disease protocol, which I will explain in the coming slides. So there are a wide array of diagnostic tools are available. The most important aspect here is that what is the most effective, economically useful and reachable to the remote place of maize field is where we focused upon. So that I will explain to you in a minute. And we focused on how we did our efforts on MLN monitoring and surveillance. And also we worked with various partners on seed companies. And also we created awareness among the various stakeholders. So these are all the various approaches we adopted to curb and prevent the spread of diseases in Eastern Africa and also prevent it movement from Eastern Africa to Southern Africa. So most of you may be knowing, or some of you are new to these diseases. Mesolithic necrosis is a viral disease, and it is caused by a combined infection of maize chlorotic mottle viruses with any one of the partyvirus. In this particular case, it is a sugarcane mosaic virus. The first report it was reported in 2011 in Kenya. Further, it spread in Uganda, Tanzania, Rwanda, Ethiopia, and then DR Congo. And of course, there are unconfirmed reports in Burundi, but there are no reports. Further, we did not see any MLN report in either Eastern Africa or Sub-Saharan Africa. So that is the major development through our efforts. So some of the information which I would like to bring your attention that in the globe, around 20%, 22% of the yield loss is mainly due to diseases. And there are very many diseases are affected in maize, and pest is also associated. So in this particular pest and disease is contributing to the tune of 22% loss. Especially in Africa, the major invasive pest or diseases happening here that and now frequently because of a continuous cropping pattern, favorable environment, and host, susceptible host, and also other related hosts and vectors. Africa has been noticing several devastating pest and diseases, examples such as Mesolithic Necrosis, and then wheat rust and fall armyworm, fusarium wilt. But in case of maize, it's MLN and fall armyworm. Dr. Hugo, in CIMIT socioeconomics, has done a wonderful survey and in understanding the economics, the part of the diseases. He has done assessment in 2016, considering the economic yield assessment. So in those period, almost 0.5 million tons of value of maize has been affected, which is to the tune of $180 million. So you can imagine 5 countries in Africa, how much yield must have been lost due to MLN. And if it hit in Southern Africa, you can imagine the tune of damage. And Southern Africa is one of the food bowl for entire Africa, where larger maize is being produced and exported elsewhere to the world. But the similar study he conducted in 2019, 2018, the disease, you can see the disease severity has been completely reduced, but still the disease has not wiped out. So this is the kind of study the same team has done. Of course, I'm one of the team member. So this information is available in this literature. Anybody can refer to this literature. As I was mentioning earlier, that Mesolithic necrosis is a combination of 2 viruses. It is not a one virus. And Mesolithic necrosis, remember, this is not a virus. This is a disease. It is a symptom. And the virus is 2 viruses in this case. And when they combined and cause the damage to the plant at early inoculated stage, especially from V5 to V10, V14 stage, the crop loss is almost 100%. The symptoms is shown in the next slide. So individually they can cause the diseases, but the damage is so severe when they are occurring in combinations. So as you all know that a viral disease having a different type of symptoms, especially the chlorotic speck and streaks, mosaic, mottling, and then drying of the leaf, especially in the premature death if it affects at early stage. And the malformed are no ears, and sometimes the rotting cups. So this is a disease which never sleeps. It is first reported in 1974 in Peru, and then USA, Argentina, Brazil, Thailand, Mexico, Hawaii, China. First time in Africa, it was reported in 2011. And latest report was DR Congo. And then it has reported in Ecuador and Taiwan and Spain. So this is the kind of a roadmap how the disease has moved from various locations. So the good news is that it is not reported in India, and I pray that it should not come to India. That is the very, very deadly diseases, which I don't want to see it in India, and also any other countries for that matter. And you may ask the questions, why this is devastating? Why is not other diseases devastating? There are few factors which are associated for this rapid spread of this disease. Maize chlorotic mottle virus is new to the region, and potentially the new strains of FCMV or MDMV. And in Africa, you may be knowing the weather is so conducive, farmers always prefer to grow throughout the year the maize because there are 2-3 reasons. One is for their food and income and livelihood. So that condition also favors. And then the lot of collateral host, alternative host is also hosting the inoculum for the viruses. Most of the varieties grown in Africa is susceptible. Nearly 98% of the commercial varieties are 100% susceptible. and wide spread cultivation of this germplasm with conducive weather and vectors and movement of susceptible seeds due to porous transboundary mechanism, which has led to the serious damage of the crop due to Mesolithic necrosis. So, especially the major outcome with many efforts, what we have noticed that we have worked in the major regions, especially in Eastern Africa. We developed this multidisciplinary, multi-institutional strategy for tackling these diseases, and which I will explain in the next slide. And we also developed 19 resistant tolerant hybrids. And due to our continuous research efforts, we converted 52 the most susceptible lines into resistant lines through MABC. That is one of the remarkable achievement in the breeding with this kind of constant interaction with pathological efforts and as well as marker, double haploid and breeding. So we also developed a diagnostic protocol and in terms of disease surveillance and diagnostics. So as I was telling you in my previous slides, it is not a one institutions, it is a multi-institution, multidisciplinary. We address these major challenges through many approaches. First is, as you all know that CIMMYT is mainly focusing on breeding, but in this case, it is not just breeding, it is multi-pronged approach. But in case of breeding, we did a very intensive work to fast track the development of resistant hybrids and release and to give it to the farmers with the help of NARS and seed companies. We also collaborated with various research organizations, NPPOs, and various American and European research institutions, universities. And later we also engaged with the NPPOs and several local international organizations for producing disease-free commercial seeds and exchange these seeds with various countries in Africa. And we developed a very intensive, rigorous disease monitoring surveillance mechanism. That is one other key milestones to understand this disease. And we also trained several small and medium enterprise companies in terms of better agronomic management practices. And also we tried to reach out many organizations in terms of communications strategies. So with these various approaches, we could tackle the diseases. That is one of the key milestones for entire CGIAR in the region. So I will just touch upon key stages, first starting with the breeding. When the disease affected, you can imagine farmer could not get any yield and their children could not go to school. They could not get their livelihood. Really, it was like it is like a corona of maize. So, but our team in CIMMYT with the various partners, we could put our efforts in such a way, a lot of available germplasm was screened, fast track mechanisms. And at that stage, I joined CIMMYT. So together with the team effort, other colleagues, we could screen several germplasms, and you could see the level of resistance. It is not just green alone; it also gives 6 to 8 tons per hectare, where in the disease pressure of highly affected maize with the sun. And with this, farmer could smile. So, as I was mentioning earlier, that we did not just screen as a just simple one. It is a public-private partnership where we supported more than 10 to 12 countries in Africa, their research organizations, more than 20 private companies, and then CGIAR. In terms of germplasm screening, so far we have screened around 215,000 germplasms, which is equivalent to more than 330,000 rows of maize. Each row is almost 3 meters and 13 plants. So majority of the germplasm was contributed to screen is CIMIT, and remaining is small and medium enterprise companies and NARS. So the quality of the data is quite good. The heritability is 0.85, be it breeding or molecular breeding, gene editing, any experiment, we could get a very high quality data. And the impact so far with this kind of efforts, we could release 19 MLN resistant or tolerant hybrids in Eastern Africa. And earlier we were having, only 5 inbred lines which are tolerant. Today with our molecular breeding team, so we could develop or identify and develop and deploy almost more than 50 lines which are resistant now, but they were highly susceptible. You can imagine this is the screening facility which you can see here, where 20-hectare facility, there are 2-3 criteria. When we develop this facility, there should not be any maize grown around this area. Number one, because any external maize disease should not come to this facility, and this disease should not spread around any other crop. So that is the precautions we should keep, because this is one of the deadly virus, and also it should not again act as a source of inoculum. And then all the movement of people, tools, implements has been restricted with a high stringent quarantine. And we don't self or cross-pollinate anything here. And third is that anything which is taken inside, which will not be given outside, either in plant materials, anything outside to from the facilities. So it is not just a screening facility. Another interesting part is that when there are some resistant hybrids, inbred lines were noticed in USA, when they were screened here, they were 100% highly susceptible, meaning that there is a need to identify, develop the resistant hybrids under these conditions, because of the virulence of these pathogens is quite aggressive here. So with this kind of aggressiveness of the disease, we could able to get this kind of clear resistance merge of susceptible hybrids. So this is entirely 20-hectare facility, out of which 3 hectares dedicated to screening facility in terms of lab, net house, which is meant for keeping the inoculum, and also to screen for individual viruses. And then the water reservoir is dedicated, highly enclosed, and we irrigate and fertigate entire field through automated drip fertigations. So thereby we limit all the variabilities due to environment and nutrition. We get a high quality of data. So it is not just a facility alone. Because of several stringent measures in terms of a very good robust inoculation protocol, we grow inoculum separately in terms of MCMV and SCMV. We harvest the leaf, and then we get it, and then we chop it, blend it, sieve it, and mix the viral inoculum 1 MCMV, 4 SCMV. And then once we mix it in chilled conditions, we put carborundum. Then we use the very power sprayer to inoculate the field. We inoculate at around 5th to 6th leaf stage. And after the 6th leaf stage, we just allow the plant to express symptoms up to 21 days. After 21 days, we score the diseases. We keep a 10 days interval for inbred lines and 14 days interval for hybrids. We score 4 times. We consider third score is the critical for judging or assessing the disease severity in terms of knowing the resistance. And in terms of facility, we do not allow our movement of the restrictors. We completely disinfect. We prevent the movement of people, allow them to disinfect using this change room facilities. We also having restrict the media facilities and net house. And through this, we developed a system. And this is, you can see the automated fertigated systems. And we use the handheld for taking the data. With this, we can completely work on knowing the diseases resistance. We also are working on using the drone in terms of disease recording. And in terms of phytosanitations, we use a different color for each viruses. This is for sugarcane mosaic virus. This is for maize chlorotic motor viruses. This is in the field. The people who visit in the field are not supposed to come to the greenhouses at any given time. Whatever the materials is been grown in the greenhouse and the field completely incinerated, not a single plant tissue will be allowed to give an out in spite of any request. So as I mentioned, this is the most important aspect you should remember, that our strategy is to screen 2 times and index 5 times. The strategy is that we should develop a resistant variety when the disease is already in the field. We should not develop a resistant varieties or hybrids once the disease is completely managed. So that is the intention, to have a 5 times screening in the entire year. So we have developed a very good disease screening protocol, 1 to 9 scale, which is 1 is clean and 9 is completely dead. This is for inbred lines and this is for hybrids. All these protocols has been clearly described in our technical manual. Anybody is interested can refer that manual. So when we started our efforts, these are all important inbred lines. They were highly used and adopted in various agroecological regions of the maize. So in various combinations. But the challenge is that they were highly susceptible to Mesolithic necrosis. And they could not get any yield. So we were very scared what to do on those days. But when we identified a resistance source in as a KS23-6, so this acts as a resistance donor for various our germplasms. Further, we developed and improved, identified the marker using this source, and also we are working on the gene editing through the collaboration of Carteva. So in a minute, you can see here how we have done the effort on this background of susceptible lines. Our breeding team, Dr. Bayane, Dr. Dan Makumbi, and various other colleagues in breeding team in Eastern Africa have screened many lines and in many years. But when they started, so many lines were susceptible, and the average mean of disease severity is almost 6.5. And when they continuously improve their level of resistance, gradually they have improved 2018 almost 5 on the 1 to 9 scale. I can say confidently today, our level of resistance somewhere around 3. It is a key milestone for entire CIMIT breeding team with the constant support from pathology team. So we could develop a resistant hybrids under artificially inoculated conditions. It is not enough to say that we have identified, because it should also perform better in other hotspots. We grow the same hybrids in a hotspot where MLN is the challenge. Still, we could get the better yield. And you can see in Tanzania and also Ethiopia, it is not resistant just in terms of greenness, but yield is also very good. That's how the farmers were very happy. When they were grown in the optimal conditions, the yield was so excellent. So that is another comparisons. So what I want to say that under disease pressure, the commercial hybrid is losing the yield almost 77%. But whereas our second generation is just 3.1% yield loss. This is the kind of a development which we could do through breeding. And so far we have released almost 19 hybrids. You can imagine this is the resistant hybrids looks like this, whereas the commercial hybrids are like this. So many people, they make questions or understanding, they have a difficulty in understanding what is resistant and tolerant susceptible. Susceptible is such that it has a severe disease pressure, it bears no yield, and whereas tolerant, it can have some level of disease severity, but the yield is almost 4 to 5 tons. If you imagine this is the resistant hybrid, this is giving 6 to 8 tons, and the disease score is almost 3. This is a kind of a comparison in terms of resistant, tolerant, susceptible hybrids. So we could bring our hybrids in comparison of resistant, tolerant. One of the key commercial hybrids is a DK777 from Bayer Crop Science. They have been doing well in the market. So far in Uganda and Kenya, the farmers are able to produce a lot of commercial certified seeds. And Kenya Seed Company is one of the key examples in Kenya, producing more than 500 metric tons. Whereas in Uganda, hybrid bazooka, they are producing almost 2,000 metric tons and distributing these seeds to farmers. So this is in terms of our conventional breeding. In terms of molecular breeding, our colleague Dr. Manjegoda has done a very remarkable work in terms of identifying the markers and also understanding the markers through through various genetic architecture of resistances, especially MCMV. And he has identified the marker and then mainly understanding the chromosomes. He could identify the chromosome 6 is one of the key regions where this marker is available. So using this KS23-6, it is coming from Thailand, Start University, which is a very good level of resistance source for our breeding program. As I was telling before, CML442 is one of the most susceptible hybrids grown everywhere. But today we could convert that susceptible line into a completely MLN resistant lines with the use of molecular backcrossing program and double applied technology. And similarly, we converted several such inbred lines using double applied lines and also conventional lines. Today, we can say proudly 52 lines are in the status of this kind of level of resistance, which is very remarkable support for especially to develop hybrids for the farming communities. So next, I will change the topic to the next topic is that safe germplasm exchange and distribution. Imagine in this region, the transboundary is very porous. The regulations are not enough to prevent the movement of contaminated seeds and grains. So we need to adopt a very rigorous steps to check the diseases. So I will just explain in a minute how we did it in CIMMYT. Mr. Tanya is in a conference call now. He is the one who is managing this facility in Harare in Zimbabwe. And this facility is established to grow our germplasm, which we ship it from Kenya. And then he first checked the presence of diseases once he received the seeds from Kenya. And he grow this entire germplasm in these facilities. If the germplasm is found, anything is having a diseases, immediately they will alert. And this is a kind of a strict quarantine facilities. I wanted to bring your attention that what is the difference between the quarantine and screening facilities. And this is also a screening facility in Naivasha, but also quarantine facilities. But whereas in Harare, it is only quarantine facility. We don't inoculate the disease by artificial means. And once we notice that is the diseases, we destroy the crop, not just in the farm, around 50 kilometers near the vicinity of this farm. And once we notice that there is no maize lethal necrosis or MCMV, then we self and give the seeds and distribute all over the world. And in this case, we do not self or cross, and we do not give seeds from this place, and it is only we inoculate and identify the resistance. This is how we have been using these two facilities for breeding and also exchange of germplasms. And for your information, so far we could test more than 10,000 composite samples in Kenya alone coming from our breeding site. And then they were tested first in the field, and the same seeds were brought into CIMMYT laboratory. We checked the seeds presence for the presence of MCMV. And once they found free or negative for MCMV or SCMV, Again, we test in KEFIS, it is a local plant inspectorate services. Once they find it free using RT-PCR, then the seeds are qualified to ship it to Zimbabwe. Once it is received in Zimbabwe, again, as I explained earlier, he will test and then grow and then sell and give the seeds to the other partners in the Africa and other parts of the world where the non-endemic regions are there. So with this kind of stringent measures, we could contain and prevent the spread of disease from Eastern Africa to Southern Africa. And it is not just these two efforts, and we also engaged in several projects on the epidemiological aspects, especially on seed, soil, and various other aspects. So I will explain in a minute how we could work on that project. Especially when we work in the disease diagnosis and surveillance project, it started during 2016. So we developed a very simple but effective diagnostic protocol. Although there are many diagnostic protocols available, like ELISA, RT-PCR, and then we could use it, but the need is only to use a very simple, effective protocol, which can be used in the field. So we adopted immunostrips, which is quite helpful. And we used simple ODK tools in the mobile phone. So that is how we optimized. And the entire protocol is been given in the technical manual, which is already available to everybody. So how we developed, standardized the protocol means anybody can do it. The simple is that we developed the protocol and we optimized. In a hectare, we could take 6 samples in the vicinity of 20 kilometers. And then we brought 6 samples and then collected, and then in the small coin size, which can be arranged in 5 minutes. And these 6 leaves can be put in a leaf extraction pouch, and this can be grounded in the next 5 minutes. And once we have this buffer ready and put it in the site with buffer, the diagnostic buffer, within 15 minutes, we get to know whether the virus is there. If there is a virus is there, you can see 2 strips. If there is no virus, it's only 1 band. If it's invalid, is no band. So within maximum of 25-30 minutes, you have the result on the spot. So this particular effort is being done in the field. Anybody in the field who are visiting in the field, they can do in just half an hour. So in a day, they can do more than 30-40 samples, and that's how we did it. And to do that kind of sampling, I was involved in training several key plant protection officers and research organizations in 8 countries for the 4 years. So with this, we developed the data points. These data points has been put it in the MLN web portal. That is, you can see in this mln.cimit.org. That web portal can give you the complete information. When you go to that web portal, when you click it here, you get to know what is the location, when the crop was planted, which variety is that, and what crop stage sample was done, who has done. All those information was used in the GPS mode. That information one can notice. So once the disease is there, it can turn into red. If there is no disease, it can turn into green. So we started in 2014. We also used some historical data from our partners. And then in 2015, the disease was so spread, you can imagine, in Rwanda, Uganda, Tanzania, and Ethiopia. So everywhere the disease has spread. And then again, 2016 alone, we could see that very many countries are showing the diseases. But touch wood, there are no disease in Malawi, Zambia, and Zimbabwe. And further, we also conducted a survey separately in 2017 and 2018. You could see Ethiopia, Rwanda, Tanzania, Kenya, they're all having diseases. So put together, you can see still there is a lot of disease there in Ethiopia, Uganda, Kenya, and Rwanda, and Tanzania. but there is no disease incidence is observed in any of these three countries. That is the good part of this effort. So what I was referring earlier, recently we published our technical manual, whatever the effort which we have been doing in terms of breeding, disease surveillance, diagnosis, producing disease-free commercial seeds, disease germplasm exchange, all these protocols, whatever we have been practicing for the last 4 to 5 years, we compiled and distributed to all our partners. This is a very comprehensive manual for all the partners, for those countries which are already using it, and also for those countries like Asia or any other countries where there is no MLN, but they can use this as a guiding document in case if they have to do it, they can simply use this manual. And also we are here to give the training in every aspects. So this is the training, just some of the data I was referring. So we did not just sample only for the growing field, but we also collected the seeds from the agri dealers, because just not the field, but also we collected samples from the agri dealers. We detected it. We know that there is a lot of seeds coming as susceptible. So we alerted all the partners and also companies. And through this, we have done a kind of a robust disease surveillance. In terms of producing MLM disease-free production, we could engage AGRA, Alliance for Green Revolution in Agriculture in Africa, and also AITF is another organization. Through their support, we could train many participants, almost 574 participants from NPPOs and NAS institutions. And also we trained several small scale contract seed growers. So together we could train them in every counties and countries. So we developed a checklist, and we optimized the checklist with the consents of various private companies and national partners. And we come to the point where this is the proper checklist to be adopted. And remember now almost 34 to 35 companies in Africa, they are using this checklist rigorously to produce disease-free MLN seeds for their local use. So we just help them in terms of giving the literatures in English, Swahili, Amharic, and Spanish, and it is available to translate to any other language as and when it is needed. So this is kind of the training literatures been distributed. More than 30,000 literatures has been distributed for the entire one. So most importantly, that with this kind of SOPs, we could be able to help institutions for proper diagnosis and also control the diseases in the endemic regions, and also prevent the spread into the non-endemic areas. So this is, again, I was just referring how we conducted the training. And also, most importantly, it is not training, we should take it as a point. It is a capacity building to every single partners, whoever are required to take up. So we trained in all these 8 countries in terms of using this ODK tool, and also how to collect samples, how to diagnose, and in the field and also in the lab. Everywhere we try to help them to take up this training. So these are all some memories or glimpses of training program in Tanzania, in Rwanda, and Ethiopia, and Kenya, and several places. The most important aspect is the community of practices. Remember, community of practice is the key for any such massive efforts which we undertake. They mainly increase the MLN knowledge and share among the shareholders, and harmonization of disease surveillance and disease diagnostic protocols, and adoption of rapid diagnostic using immunostrips by seed companies. And you know, like when the COP developed, it is the key objective is to seek an agreement on the phytosanitary practices on the region. And second, the cooperation is quite important among the partners. That is where we steer the team. And we also help them to share what is their learning and share across the borders without any hesitations. And also we strengthen the linkages and provide opportunity for the collaborators to exchange their knowledge and constantly report on the progress, whatever we have done. And finally, provide an information, all this in the MLN web portal. So it is not enough. We also engaged the policy holders, especially East Africa community and several seed company like Stack, several such key stakeholders we trained and also we alerted them and also enabled and influenced them to make some policy changes. So all this information, what is being discussed now available in MLN portal, he is Dr. Prasanna. I think you're all familiar with him, and he is our leader. He has supported to take up the entire task, not just this one, and also now he's a leader for plant health initiatives in 1CGIR for all the crops, all the pests and diseases. He's Director General of Kenya Agriculture Research Organizations. He's also coming and regularly in touch with the program. As I was explaining to you earlier, that we just did not focus on a few aspects, but extensively we're also working on the gene editing, especially through the collaboration of Carteva seed companies. And the key aspect here is that in this kind of critical projects, the plant score was done in one plant-to-plant basis. Even at that level, high disease screening data has been established and provided. Now, almost the project is in a final stage. Perhaps very soon we can also come up with gene-edited resistant hybrids. Yeah. So this is how the progress has been made. In summary, Mesolithic Necrosis is a disease in Eastern Africa, which is under control through constant surveillance and better quarantine measures and a host-free period of 2 months and producing the MLN disease-free seeds. Southern Africa is maintained as a free of MLN and safe exchange and for MLN free seeds, and emergency preparedness is also available, and that is how we focus on. And then the focus on resistance breeding is still on the upfront. We are working on it and use of various technologies. And then we also constantly support our partners. And one of the review article has been published last year in the virus research. Whatever I have discussed and mentioned in the presentation and also available, it is well summarized in this manuscript. You can refer this one. So with this, I would sincerely thank all the donors, especially USAID, Bill and Melinda Gates Foundation, Syngenta Foundation, CRP Maize, and various other partners. And we immensely thankful to Kalro for providing such a wonderful facility in Kenya. Without their support, we could not have been done such a fast progress to the farmers of Africa and also support our private seed companies. And especially, I immensely thankful to all the CIMMYT colleagues where I was so happy, and then I could contribute whatever little I can do for the maize. With this, I would sincerely thank the organizers for giving me an opportunity to share this information. Remember the technical manual, what I mentioned, it is available and all the chapters are well described. Anybody who is interested to get, it is available, I can share. And also I am available for any discussion now. Thank you for your interest and your patience listening. And yeah, thank you again.
Thank you very much, sir. It was such an excellent lecture. It was really an extensive work to understand the disease as well as to come out with as many as 19 resistant varieties. Great service to the farmers, sir. Thank you so much. Now I request, now platform is open for discussion. Now I request if somebody is having anybody.
I think Dr. Ravi Khetrapal has raised his hands. Dr.
Ravi Khetrapal, sir, please.
Thank you, Dr. Prashant, Dr. Pratibha. Well, Dr. Suresh, your presentation was like music to the ear. We really enjoyed it from the angle of doing something from the very beginning to the end, like starting right from detection to resistance to going up to community of practices and capacity building. really gives a very massive and comprehensive effort made to manage maize lethal necrosis. Not a question, in fact, I just want to know, which probably I'm ignorant, since you mentioned resistance is very important, and that is the best way of managing plant disease, we all know. But there is also a way which boomerangs on plant pathologies because of resistance. We have new strains and new biotypes which keep on coming. So resistance has its dual face, but very important. Now, in case of seed-transmitted viruses, like here, most of the resistance are recessive and monogenic. I do not know about this maize lethal necrosis. What is the actual resistance level? Because recessive and monogenic resistance normally are more easily overcome than the polygenic, which we have against fungal diseases and all. The majority are polygenic. So how robust is our resistant program for the long run, I'm asking you. I may like to know from you. And secondly, you have given a good account of work done on detection also. And are we, because maize is a monocot. So do we have an idea on the location of the virus in the seed so that we can have more targeted detection? This I'm not clear. And my third point is, since you have also been working with the private companies and all, and even in the quality control, I mean, you means the whole program, which is excellent. I would like to know if you have worked on tolerance limit, like what level of the infection in the seed can lead to an epidemic in the field. Is it 1%, like it is 0.001% in case of lettuce mosaic virus, or 0.1 or 0.2% in case of PC-borne mosaic virus? So what is the level of tolerance? Tolerance limit is very important for seed certification. Do we have those information? Because those are very vital for this very successful program to remain successful. Over to you, Dr. Suresh.
First of all, it is a really honor for me, sir, to have you in the lecture, and it's a great pleasure. And also, I'm very thankful for those aspects of the questions or comment, whatever you advised. I fully agree with you. These are all very important, relevant questions I must have addressed to the presentations. Number one, I will touch upon the threshold. The threshold of seed transmission is the key, in which KEPHIS, Kenya Plant Health Inspectorate, they have started with the 10%. Then once the notice 10% is too high to adopt the threshold, and then they brought it to 1%. And 1% itself is too high for producing disease-free seeds, especially in private seed companies. Now they adopted 0% tolerance, means a plant is noticed in any field, it is completely rejected. And that is where the stringent measures has been adopted, be it multinational, be it small and medium enterprises, be it government organizations. They carefully diagnose the field, and we have very good respect to them. And not one time, 2-3 times, and they come up with the 2-3 critical stages. A plant has been identified is completely rejected. Imagine, I personally have seen in a private company more than 500 metric tons of seeds is piled up due to rejections. And with that kind of stringent measures only, the production of disease-free seeds, especially for commercial seeds, for managing MLN is the key. That is an important question. And we are also working on some sort of a chemical seed treatment, whereby, like there are other crops which they are using chemical seed treatment for especially viruses. So whereby we can minimize, completely eradicate that kind of infections. That is another part of it. But as far as is this is another part, as you mentioned upon, that disease resistant tolerance, when we describe our hybrids, initially when we developed first generation, it was tolerant 1 to 9 scale, it was 4 to 5 scale. Then we realized that itself is not enough. And through our constant use of innovative tools and technologies, we developed hybrids to the tune of 1 to 9 scale, 3 scale, and which is giving 6 to 8 tons per hectare. And this particular mesolithic necrosis is affected by 2 major viruses, that is MCMV and SCMV. It's polygenic recessive in nature. So, and that is most important. And also we agree with you, it is not enough to identify under only artificial conditions. We need to constantly monitor our hybrids in various hotspots to see whether this hybrids has been affected various other associated viruses or virulent strain of the same viruses, so that we can be sustainable to support our communities in terms of giving the resistant hybrids through various approaches like developing a sentinel plots in terms of monitoring our resistance program. I hope I have given some of the information to your questions or comments. Perhaps I will include these points in my future presentation, but it is very, very important. Thank you, sir.
Thank you, Dr. Suresh. In fact, this mesolithic necrosis will remain as an example for how to manage a disease and how a proper surveillance and monitoring and resistance program can really help in managing. Why I'm saying so, because I wish it never comes in India and other parts of Asia where it is not there. What we need is a very prophylactic measure to see that it doesn't go where it is not there. That is very crucial. And I'm sure Dr. Celia and people in quarantine must be aware of whatever we receive from CIMMYT as germplasm will be surely free. It comes from a very good germplasm handling unit. But whatever comes from other labs of Africa, I don't think if we have a control. Like what CIMMYT is doing is par excellence. I'm a partner in your CIMMYT's global program, a plant health initiative by Prasanna and all. And recently we had a meeting on like how this 1CG plant health initiative we can bring in Asia, Asian part, because they had different chapters across the world. And my major point was how to avoid disease, and they have listed mesolithic necrosis is one of the 22 major pests which has been shortlisted, which might change. But the major point is how we can avoid it. Avoidance, and the best principle is resistance. That is number one. And secondly, of course, avoidance is by the prophylactic timely detection and management. So avoidance principle has to be fully implemented, and the message has to go to countries which have maize but which do not have MLN. You know, that is very crucial. Where it is there, there is CIMMYT. People like Dr. Suresh and many who are really fully involved to manage it. Because avoidance will really help. And this is not only maize. I see this management program as an example to be followed at a global level, especially in Asia and Latin America also, how to really tackle such a serious problem, which is seed transmitted. I think from that angle, it is a success story. I mean, it's a global success story.
Yeah.
Thank you so much,
Thank you, sir. And
all the best.
Yeah, only another point which I might want to contribute to this discussion, that there are many partners from many countries. They came forward as a proactive breeding approach, though the disease is not reported, but like Rasi seed and several other partners in Asia, they came forward. Further, they want to develop their hybrids as MLN resistance through our novel technologies. And before the disease emerges, at least they have some basic solution. Although the prevention through use of disease-free seeds. But this is another positive approach has been not.
Thank you.
Dr. Celia.
Thank you, IPS, sir. Thank you, IPS, for this opportunity. I have to jump out for another meeting, so I'm sorry I'm leaving you in between. All the best, Dr. Suresh. Just keep it up.
Thank you.
Thank you, sir, for your presence.
Thanks, Dr.
My
Ketipal
pleasure.
and Dr. Alan Suresh both. Really.
Thank
for
you so much,
joining us.
Pratibha.
Dr. Ravi is always, like he has always participated in our talk series. Thank you, Dr. Sir. Thank you.
Thank you, Dr. Pratibha. Thank you, Dr. Prashant. Thanks,
Thank you,
everyone.
sir. Thank you. Dr. Celia, ma'am, do you have any question?
Yeah, Dr. Suresh, if you unshare the screen, that will be good.
Okay, just a second.
And thank you very much, Dr. Suresh, for a very comprehensive presentation, including the quarantine measures that are being taken up. And I had the pleasure and I listened to him in many other conferences, including the International Plant Virus Epidemiology Conference. And you have we in quarantine actually intercepted maize chlorotic mottle virus in germplasm imported from USA and one or more countries. So you mentioned instead of seed transmission, seed contamination in one of your slides. And also the seed transmission of maize chlorotic mottle virus was very low percentage, 0.04%. Even then, it is very important it has been reported in 1990s. If we look at the literature, if embryo is infected, then it is a major quarantine importance. What is your comments? Well, I want to know actually the contamination part and also low level of seed transmission of maize chlorotic mottle virus, not the complex. And since you have mentioned that you are testing the seeds, are you testing the whole seed or you are testing the embryo?
Okay. First of all, thank you, Cecilia Madam, for your constant motivation for this kind of interactions. Wherever I think you always want to have this topic is one of the key examples. For that, I'm very thankful to you. Coming to the point, the percent transmissions. Recently, one of the manuscript was published by Kimani et al. Through our collaborators. It is almost 0.089%. You can say 0.1%.
Yeah.
So like if you calculate 0.1% of seed transmitted plants in per hectare, it is coming almost to 6 to 7 plants. It's quite high, which is not acceptable. That's where our KFIS Kenya, they are being rejecting the lots. So which is coming mainly from the embryo to that point where the seed virus is there and it can reflect in the seedlings when it is grown under a controlled condition without any external contaminations. The contamination part is coming is another source of inoculum where without embryo also it can stay on the maternal tissue on the seed, which is acts as another additional source of inoculum. So that could be a very high tune. A contamination could be a very entire 1 kilo of seed can be contaminations. There is no percentage. It depends on how the seed handling after harvest is been handled, whether they have been able to remove the infected plants or clean plants, remove the infected plants and keep the clean plants separate. And that is very challenging. And the third part is also sometimes the people use the grains, contaminated grains. So in all these cases is not permittable. And also I would wish that any 1.01% contamination is also highly contamination should completely reject, incinerate. That is the big source of inoculum for a country like India. It should not be acceptable, and be it any level, it should be rejected and incinerated.
Yeah, for quarantine purpose, it is 0%. Tolerance is 0%. Even if we see one plant also, we incinerate that. Yeah. I'm still not clear about the contamination part. Do you mean by contamination for seed coat infection? What do you mean?
Yeah, seed coat and maternal tissues where it is attaching from the ear to the seed. The small maternal tissue is there. That is the place where the virus is deposited there. That tissue is almost having viral component, not a seed coat.
Okay, thank you. Thank you, Dr. Suresh.
Yeah. Perhaps I will
Yes,
include that type of small picture in the next presentation. That is the most important aspect to clarify.
Yes, yes, especially
Yes.
for quarantine purposes.
Yeah.
Yeah, Dr. Prasant, may I ask you some question?
Yes,
to
please, sir.
Dr.
Yes,
Suresh?
please. You are maize pathologist. Dr. Huda is there. You must answer that.
So, Dr. Suresh, I am thankful to you because you accepted to deliver your nice experience through the lecture. So, I have read your article, which was published in Virus Research. The moment it was published, it was circulated like hotcake, that article. I remember. And just I want to know, because you have just projected that you have developed diagnostic kit like lateral flow device. This while diagnosing, so the detection level, how much percentage success, whether false positive, false negative, also sometimes traveling, noising.
Yeah. First of all, thank you, Dr. Bugai, for giving this opportunity and also be with me always to support this kind of work to be interacted. And thank you. And coming to this particular question, is a very important question as we use the immunostrips, which is a lateral flow, you know it. And we adapted the, first of all, the company say that small leaf tissues. And when we try to adapt that protocol, we should also look for economics, and especially on 8 countries in different agroecologies at a very small scale. So we tried to bring the 6 small leaf discs, and when we use 6 leaf discs, and then we diagnose the infected leaf disc and also healthy leaf disc and also some checks. So we could not have any false positive. And also second thing is that basically it is a simple diagnostic protocol in the field. In case there are second and third step is there, it is not, we don't advise for the seeds. We mainly advise for leaf tissues. In case in the non-endemic country, in case the first stage, if it is found positive, we should not just stay on that spot. Then we take those samples for ELISA and RT-PCR. But this is surveillance mechanism. This tools is developed, and then it is basically from Biorip or Agdia, which is commercially available. But we are not advocating as a company for a commercial purposes, but we only promote the technology. But that works very well for disease surveillance mechanisms. But in places where non-endemic countries, we do take those samples for ELISA and also for RT-PCR for further confirmation.
Okay, thank you, very nice, very good.
Yeah.
I just want to add, Dr. Suresh, I'm not promoting or advocating to take the kits from Agdia, but when compared to many other commercial kits, the ones we get from Agdia is more reliable.
Yeah, I fully echo what you said, but only we put a disclaimer. Yeah.
Telling unofficially.
Dr. Pratibha, ma'am, do you have few words to say?
Yeah, I really thank Dr. Suresh for really giving such an excellent talk, and it made everybody, discuss so many points, and especially the people who are working with maize pathology, Dr. Gogoi and Dr. Ravi and Dr. Celia, because they are connected with the quarantine. So every time very good questions and very good interactions take place. Thanks, Dr. Suresh, for all the discussions. This can continue, the discussions can continue, but I think now the time is coming. We have to close down this thing. So only what I can give is I thank Dr. Suresh for giving such an excellent talk. Thank you very much. No questions.
Thank you, ma'am. Now, coming to the end of the session, I will request Dr. B. Parmeshwari, Senior Scientist, NBPGR, Hyderabad, to present vote of thanks. Dr. Parmeshwari.
On behalf of Indian Phytopathological Society, I feel immense pleasure to propose vote of thanks. At the outset, I extend my profuse thanks to Dr. L.M. Suresh, Maize Pathology Lead, Sub-Saharan Africa, International Maize and Wheat Improvement Center, Nairobi, Kenya, for accepting our invitation and delivered enlightened lecture on maize lethal necrosis, effort towards containing the spread and impact of a devastating transboundary disease in sub-Saharan Africa. And also thanks to the President Dr. Pratibha Sharma Madam, Secretary Robin Gogoi Sir, and Joint Secretary Kalyan K. Mandal Sir, and organizing committee members, and Celia Madam, Ravi Ketrapal Sir, and others for asking and participating in discussion. Then organizing committee members Prasant Jambulkar, myself and Suresh, Pramod Kumar Gupta, and G. Prakash, and Sangeeta, Malkan Singh Gujar, and Dr. Sushil Kumar Sarma for commemorating this Diamond Jubilee lecture series. And also my special thanks to all the participants and learners who attended this lecture series and got benefited. Thank you, one and all.
Thank you,
sir.
Parmeshwari.
Thank you.
Thank you.
Thank you.
Namaste.